Good morning, student. Today we'll discuss biotechnology and its application. That is chapter number 12, and we have to learn about some genetically modified plants as well as the application of biotechnology, how we can get the benefit by applying the biotechnology by recombinant, we can say the DNA, by using the genetical engineering. So all these things we have to learn in this chapter. Okay, so here you can see biotechnology. It is actually applicable to chemical industry, it is applicable to medicine, agriculture, power industry, environmental protection, food industry. So huge applications are there. So now we are coming for the introduction here. Biotechnology deals with industrial scale production of biopharmaceuticals, means medicine and their field, and biological using genetically modified microbes, fungi, plants and animals. So we are using this biotechnology in medical field as well as the, we can say, therapeutics, diagnostics. In that way we can use that one, and as well as we are using the genetically modified plants as well as animals, microbes, fungi. So we are using these things in biotechnology application. Applications of biotechnology includes, what are they includes? Therapeutics, diagnostics, genetically modified crops for agriculture, processed food, bioremediation, and waste treatment and energy production. So these are so many fields are there where we can apply the biotechnology. Okay, next we are coming for major research areas of biotechnology where we can where we should go for researching, we can say the field, to supply the best catalyst, to supply which one, to supply the best catalyst and microbe pure enzyme in the form of improved organism with better characteristics, to create the optimal conditions by genetic engineering for a catalyst to function, means what actually the optimal conditions are needed. By genetic engineering we create that one to catalyst to function, and purification of protein organic compound by developing the downstream technology. Already have learned last class, last chapter, what is the downstream technology, the purification and separation of the desired product, that is called the downstream technology. So main research area is for best catalyst, supply the best catalyst in the form of microbe or pure enzyme, and to create the optimal condition by genetical engineering, that is for to catalyst to function. Not only create the supply the best catalyst but also create the optimal conditions, and purification and separation, that is actually called the downstream technology. Okay, next one we are coming for बहुत ही इंटरेस्टिंग जो है इधर, बायोटेक्नोलॉजिकल एप्लीकेशन इन एग्रीकल्चर, how we can make use of biotechnology in the agricultural field. So first one is the what is coming here, increase the food production, can be achieved by agrobiochemical based, means it is increasing the food production by which one, by agrochemical based agriculture, means agriculture as well as the chemical conditions are applying there, organic culture where no chemicals are used there, genetically engineered crop based agriculture. तो हम ये देखे हैं कि आइदर मीन्स फूड प्रोडक्शन को इंक्रीज हम कैसे कर सकते हैं ना, एग्रीकल्चर फील्ड में हम फर्टिलाइजर को यूज़ करना, पेस्टिसाइड को यूज़ करना, that is agrochemical based agriculture. Organic culture means where we are not using any type of chemical substance there, we are using all the living, means nitrogen fixation, fertilizer, pesticide, what we are using, all are using an organic nature or organic form. And genetically engineered crop based agriculture means third point is genetically engineered crop based agriculture. Okay, so we are coming about the one by one. So we can see green revolution, crop production increased by using improved crop varieties. What are the conditions we need? Now one is improved crop varieties, better crop management practices and use of agrochemical, that is fertilizers and pesticide. First one is the crop variety को आपको select करनी है, those who are giving the best product. And better crop management practice means in case of the we can say the crop rotation, we can say the crop जो management practices बहुत सारा है, that also you have to apply there. And use of agrochemicals, that is fertilizers and pesticide. So we can say the intercropping system, and we can say the mixed cropping system, which one is beneficial, which one is actually applicable on the basis of the that surrounding, that we have to select. That is the actually main motto of increased we can say the food production. आपको ये जब एग्रोकेमिकल फर्टिलाइजर्स और पेस्टिसाइड्स इसको यूज़ करना है, बेस्ट क्रॉप वेराइटी को यूज़ करना है, बेटर क्रॉप मैनेजमेंट एंड प्रैक्टिस को अप्लाई करनी है, दिस आर द एक्चुअली मोटो ऑफ ग्रीन रिवोल्यूशन और द वी कैन से स्ट्रैटेजी ऑफ ग्रीन रिवोल्यूशन. नेक्स्ट, फूड सप्लाई वाज ट्रिपल्ड बट वाज नॉट एनफ फॉर ग्रोइंग पॉपुलेशन. तो फूड सप्लाई तो इंक्रीज हुआ बट इट इज़ नॉट एनफ फॉर द ग्रोइंग पॉपुलेशन के अकॉर्डिंग इट इज़ नॉट, demand is not fulfilled there. Agrochemicals were too expensive for farmers to afford. Means what are the we can say the fertilizers and pesticides are becoming very expensive, so that farmers cannot afford it. ये problem आ रहा था. Increase in yield with existing varieties are not possible with conventional breeding. Conventional breeding means जो normally which are actually गोइंग ऑन ब्रीडिंग. कन्वेंशनल मीन्स ट्रेडिशनल ब्रीडिंग जो हो रहे हैं उसमें इंक्रीज इन यील्ड with the existing varieties are not possible. So these are the drawback, we can say, problems we can say it came when we see that one कि food supply तो increase or triple but it is not providing, or it is not actually fulfilling the demand of the growing population. Agrochemicals बहुत expensive हो जाते हैं for farmers to afford, and we can say the increase in yield with the existing varieties are not we can say possible with conventional breeding. Okay, next one we are coming for the introduction of. Now what is the solution? अभी तीनों में तो आपने एग्रोकेमिकल भी इंट्रोड्यूस करा है, एग्रोकेमिकल भी एक्सपेंसिव हो रहे हैं. जो कन्वेंशनल ब्रीडिंग सिस्टम इसमें भी we are not getting the highly we can say the yielding product, or the crop plants we are not getting that one. And crop production to food production increased है, that then also they cannot fulfill the demand of the increasing population. So what is the solution then? If the problems are arising, solution must be there. What is the solution then? Solution is here. Solution is introduction of first solution is introduction of genetically modified crop was an alternative to obtain maximum yield by understanding genetics of the crop. So one and only solution is what is that? Now we have to introduce the genetically modified crops. Some useful aspects of GM plants means genetically modified plants in agriculture. What are the aspects we are getting? The improvement in nutritional quality. Example कौन सा? Vitamin A enriched rice. We can get the improved nutritional quality crop. Second one, better nitrogen fixation means plants can fix nitrogen in a better way. Tolerant to abiotic stress, that means cold, drought, salt, heat, means by this genetically modified plant we can get this type of benefit. We can say we can get the tolerant plants with those who are those who can tolerate the abiotic stress. Reduce reliance on chemical pesticides. तो दैट इज पेस्ट रेजिस्टेंट क्रॉप वी आर यूजिंग. जब पेस्ट रेजिस्टेंट क्रॉप हम यूज़ करते हैं दैट विल डिस्कस, है ना. इन दैट कंडीशन वी कैन रिड्यूस द chemical pesticides, application of the chemical pesticides. Reduce the post harvest losses. What are the post harvest losses we are getting? That also can be reduced by introducing genetically modified plant. And next one is the increase efficiency of mineral usage by plant, means that prevent the exhaustion of fertility of the soil. Mineral fully utilized हो सकता है plants के through. That is the genetically modified plant की we can say the benefit. Production of disease resistant crop. We can produce the disease resistant crop by genetically modified plant. And micro propagation for biomass energy production. So these are the 8 point, and these are the useful aspects of GM plants, means genetically modified plants in agriculture. Okay, let's now start about the some applications are here. Now pest, today we'll discuss about the pest resistant plant, how we can create the pest resistant plant, that is also genetically modified plant, how we can create that one. So look at the first one, what is coming now. Now we can see here a pest resistant plant, how we can create. First one, biotechnology in agriculture pest resistant plant, development of pest resistant plants, reduce the use of pesticides. Okay, so if we are using the pest resistant plants, so what we can use, what we can reduce the use of pesticide, we can use one such approach, introduction of bt toxin, means this is actually we can say BT means Bacillus thuringiensis. This is a bacteria, and they are producing the BT gene, actually cry gene, we can say. So that is the BT toxin is produced, and that is actually introduced into the target plant. Okay, so BT example, what is that? BT cotton, BT corn, rice, tomato, potato, soybean, etc. So why we are introducing this BT toxin gene in target plant? That is the story is now. BT toxin produced by Bacillus thuringiensis due to presence of BT toxin gene. ओके ये जो टॉक्सिन होता है दैट इज प्रोड्यूस्ड बाय वन बैक्टीरिया दैट इज कॉल्ड बैसिलस thuringiensis, and who is producing inside this bacteria, one gene is there, bt toxin gene, that is producing this bt toxin. Toxin means a substance which can create the toxicity. BT toxin gene cloned from the bacteria and expressed in plants to provide the resistance to insect. No need of insecticide or pesticide. So there is no need of applying the insecticide or pesticide in the field. Rather, if we include or introduce this BT toxin gene by cloning from the bacteria and introducing into the plants, we can provide the resistance to the insect or resistance to the insect of that plant, means that plant now is ready or resistant to that insect. So that is the main thing, that gene we are introducing into the plant by extracting from the bacteria, and we are introducing by using the same technique there, same genetical engineering technique there, and then that gene is expressed in the plant, and now they are free from and they become resistant to that insect. What that insect could create the, we can say, the damage to the plant, but now when the plant become resistant, so now the plant is free from that insect. Now how it is happening, what is the process, we are now coming. So now we are coming mechanism. Okay, so what is the mechanism here? Now Bacillus thuringiensis, that is BT toxin protein which kill the insect. Like what are the insects are target here? Lepidopterans, जैसे tobacco, birdworm, armyworm, lepidopterans they are called. Coleopterans जैसे beetles होता है, and diphterians, that is flies or mosquito. So who are the target insect here? Lepidopterans, coleopterans, and diphterians. BT toxin gene expresses itself protein crystal at particular phase of the bacteria, that is called insecticidal protein. So they are creating the protein, and BT toxin exists as inactive protoxin and on ingestion of it by insect. So when inside the bacteria they are becoming inactive, but when that is ingested by bacteria, that by insect, lepidopteran भी हो सकते हैं, कोलियोप्टेरन भी हो सकता है, diphterians also may be there. See, if they ingest it, so that toxin which was inactive in the bacteria, it will become active in alkaline pH of the gut of that insect. So when this toxin enter in the body of the insect, so inside their gut the alkaline pH is present there, that solubilize the crystal and the crystal become active now, means toxicity starts now. So activated toxin binds to the surface of the mid-gut epithelial cell and create pores, swelling, and lysis, death of the insect. When the toxin become active. So they binds to the midgut epithelial cell of the insect and create pores and lysis and swelling, and definitely as a result the insect will die. So Bt toxin isolated from bacteria coded by gene cry, crystallizing protein, that's what is called cry, and different types that is called incorporated into crop plants depending on the type of crop and target pest. जैसे कॉटन बॉलवर्म के कौन सा जीन विल बी इंट्रोड्यूस देयर ना, क्राई 1AC, क्राई 2AB. They are applicable for cotton plant and the insect target insect is bollworm, and cry 1AB फॉर कॉर्न प्लांट, कॉर्न बोरार के लिए एप्लीकेबल है. तो ये डिफरेंट टाइप्स ऑफ क्राइजेंस आर अवेलेबल एंड इट डिपेंड्स अपन व्हिच वन वी शुड सिलेक्ट एंड व्हिच वन वी शुड incorporate in the plants depending on the plant as well as the target pest. Okay, so these are the actually genes are here: cry 1AC, cry 2AB, that is for cotton bollworm, and cry 1AB for corn borer. Next one we are coming for, look at here, the figure is very important, figure is here. So what is the figure here? Now first one, this is the bacteria from where we can isolate the cry gene, that is the DNA extraction and isolation. Now this is the actually target gene here, means that we desire, because this can produce the, we can say this is the cry gene, this can produce the toxic product. Next one, the cloning and designing genes में now it is incorporated, जैसे recombinant vector के साथ incorporate किया, recombinant DNA बनाया, and then it is transformed to the cell, plant cell, and plant cell in culture, tissue culture. By tissue culture we are creating the plant. This is called genetically modified plant. This is genetically modified plant, and they are carrying the gene which is responsible for creating the toxic effect. BT toxin produce कर सकते हैं. तो definitely इसको when the insect will come and feed on this plant, so definitely this gene will introduce, or that crystal which is actually produced by the gene, so that will enter in the gut and that will make the we can say the insect die. So now we are coming next one. So now we can see what is coming. Now BT gene, BT means Bacillus thuringiensis. A BT gene is transferred from the bacillus into the corn plant. It is transferred by the same process, and now the European corn borer feeds on the corn plant and ingests the protein encoded by the BT gene. That protein encoded by that protein they ingest, and then it is coming the that BT protein penetrate and collapse the cells lining the gut and the insect die. So that is the procedure when they are taking the gene, or we can say the protein which is encoded by that BT gene. So definitely that after getting entry into the that insect body, in the gut the alkaline solution is there. So definitely in presence of this alkaline solution of the gut, they become active. That crystal become active and they will make the pore in that midgut portion swollen, and lysis condition is going on, so the insect will die. This is the main motto, or you can say the genetically modified plant. So up to this we have discussed, one part is over now. So how the BT toxin plant or BT cotton, BT rice, how they are created and how they can without applying any type of chemical fertilizer, we can make the plant resistant to that of target insect there. This is the main motto of genetical, we can say the biotechnology, genetical engineering and its application.